Let's go quickly over the sense of olfaction and gustation, which are smell and taste respectively. So these are part of our 5 special senses. Now remember, these 5 special senses are all located in special sense organs in the head. They have quite a bit more complex sensory pathways and processing pathways than do the somatic senses. And they will be things like vision, hearing in equilibrium, smell, and taste. And they each have their own special area of the cortex to process these sensations. All right, so let's first talk about smell. So smell has olfactory, basically the olfactory organ is within the nasal epithelium. And it's got the epithelial layer, and as we know, we've got with any mucosa, this is a mucosa, we have a lamina propria underneath it. So we have the olfactory epithelium itself and the underlying supporting structures that are areolar connective tissues. So we have our olfactory receptors, which are these specialized cells that will detect odorants that are in the air. And then we have the supporting cells, which include some stem cells, some basal cells that will produce new olfactory epithelial cells. So this is important because olfaction is constantly regenerating the receptors, and we'll find the same is true with gustation. So these receptors will wear out fairly quickly. So when we look at the olfactory epithelium, we have our specialized olfactory receptors, and we have our stem cells and our supporting cells. We will also see we have some mucus glands that are going to empty out through the olfactory epithelium. A lot of them will be originating in the lamina propria, which is that areolar connective tissue that contains blood vessels, nerves, and some glands. So here's a picture of it. Here we see the olfactory receptors will be specialized neurons, and the dendrites of which are the actual receptors. And their axons will bundle together into these olfactory nerves, which will penetrate the cribriform plate of the ethmoid bone here and will synapse in the olfactory bulb. So remember our olfactory nerve 1, those are these here. These are our cranial nerve 1. And from there, the olfactory sensations will be relayed along the olfactory tract to the olfactory cortex. So if we look at the cells, we can see that these are our specialized cells. Notice that their dendrites are the actual receptors, and they will have receptors that bind with these odorant molecules. We have membrane receptors, I should say, that will bind with these odorant molecules. These are things that are dissolved in air that you'll come in contact with when you bring in air that contains these things. So in order for you to smell something, you actually have to have a molecule of that thing bind as a ligand to a membrane-bound receptor on one of these specialized olfactory cells. We've got our supporting cells here, our basal cells. We've got the axons of the olfactory cells. This one's a developing one. Notice its dendrites haven't yet reached the olfactory epithelium, the apical surface. Here is a mature olfactory cell, and notice all their neurons are going to bundle together into these olfactory nerve fibers, and these are going to be what perforate through, or perforate, I guess, through the perforations in the cribriform plate. All right, so the olfactory glands are going to coat the surfaces of the olfactory organ with mucins, and this is going to combine with water to form mucus. And this is going to be important because this is what's going to dissolve the odorants. So we got to have the odorants dissolved in something for them in order for them to reach our olfactory receptors. And these are highly modified neurons. So these are going to be detecting dissolved chemicals as they interact with the odorant binding proteins. So we've already talked about this. We've got our 20 or more olfactory axons that are coming together and go through the cribriform plate of the ethmoid to reach the olfactory bulb. So we've talked about that. So we've talked about this. Now, what we haven't talked about yet is the fact that we can distinguish thousands of different chemical stimuli. So basically, we're going to have cells in the central nervous system that are going to interpret the smell, and kind of like what we saw with colors, we're going to have a differential activation of populations of these olfactory receptors. One thing that we will see is that all of these receptors are G protein mediated. We also have already discussed that they do turn over, so you'll be replacing these cells often, but as you get older, you don't replace them as quickly, and they decline with age. So people who get older often don't smell things as well. All right, so here's what happens. We have the odorant molecule is going to bind and activate a membrane-bound receptor, and that's going to depolarize the cell. And in this case, the receptor is the dendrites of the molecule, or sorry, of the neuron here. So it's actually a neuron. The neuron is the receptor. And so what's going to happen is the stimulus is going to create a generator potential within the neuron. And when that stimulus reaches threshold, we'll start to produce action potentials. Now, here's how it works. As we've said, these are all G protein mediated. So here we're going to have our odorant molecule binding a receptor protein, a membrane-bound receptor. That is going to activate a G protein, which will then, in this case, activate adenylate cyclase to convert ATP into cyclic AMP. Now, the cyclic AMP will then go and bind the sodium channels from the inside and open them from the inside. And now our ions can enter. So this will now depolarize the cell. So if we have sufficient depolarization, then we'll create an action potential. Gustation will be another sense that is sensitive to chemicals, dissolved chemicals. In this case, they will be dissolved in foods and liquids that we consume. And the receptor for these will be taste receptors or gustatory receptors that are separate from neurons. So they're going to be separate receptors located in a special area called a taste bud that we'll find distributed all over the tongue and part of the throat as well. And the taste buds, we will see, will contain several of these gustation receptors, and the taste buds will have different forms. We'll have 4 different types of taste buds that we'll see. A lot of them are going to be located in the tongue, in an area called lingual papillae. And these lingual papillae are these mounds that everybody has, these little tiny mounds on the surface of the tongue. In some animals, they're quite prominent. For example, with cats, if you've ever had a cat lick you and you realize their tongue feels like sandpaper, it's due to the lingual papillae. So we have 4 types of lingual papillae. So we've got filiform or filiform, however you want to say that, papillae, and these are just for traction, for grip. And this will allow us to get hold of the food, so to speak, with our tongue so that we can move it around and manipulate it. We've got fungiform papillae, so named because they look like little mushrooms, and they have about 5 taste buds each. We have velate papillae, which form an inverted V shape on the back of the tongue, and there are usually 10 to 12 of those that each contain about 100 taste buds. And then we have foliate papillae that are like leaves. Think of the folia of the cerebellum and how it made the folded leaf-like pattern like pages in a book. Well, these are located along the side of the tongue. We will see that taste buds are these collections of cells of gustatory receptors and their supporting cells. We're going to have basal cells, which are stem cells once again, and then we're going to have the actual gustatory receptor cells. And these are going to have specialized microvilli that we're going to call taste hairs because they look kind of like hairs protruding out from the apical surface of the cell, and they will project through a taste pore. So within the taste bud, these receptors are very well protected, but of course they have to have a means by which their sensory part can interact with the solutions that we've consumed. These are very short-lived, so they only survive about 10 days before they're replaced. All right, here's a picture of the lingual papillae. Here is our velate papillae back here. We've got fungiform papillae all over the tongue. We've got foliate papillae here on the side of the tongue. And we could look at sort of the different forms of them. Here's a filiform for traction only, for gripping food. Here's a fungiform papilla with taste buds here. A foliate papilla with taste buds here. And this shows one sort of close up here. So this is a velate papilla, and you can see that it has quite a few taste buds projecting off the side of it. And then, of course, the axons will then project into this structure and bundle together into some axons that will then carry this information via our cranial nerve 7 and 9. So what are we going to look at here? Here we've got our transitional cell. This is a supporting cell. Here is our actual gustatory cell. So here we have our microvilli, our taste hairs projecting out of the taste pore. And here we have about 5 of these things projecting out here, these cells. So we got 5 of these cells with their taste pores or taste hairs projecting out of the taste pore. Here we see some basal cells, and these will produce new gustatory cells. here we have the dendrites of the neurons that will monitor these cells. So we've got 4 primary taste sensations: sweet, sour, salty, bitter. Sweet, sour, salty, bitter. So sweet's associated with sugars, carbohydrates, salty with sodium chloride, and sometimes people can substitute potassium chloride. Sour is usually an acid taste. Bitter can be basic or quinine, I think, is a bitter taste as well. We will also find umami, which is an amino acid taste. So we get this from broths and small amino acids and peptides that we'll find, and some nucleotides will trigger the umami. And this is one reason monosodium glutamate is popular in certain types of cuisines, because it will bring out tastes, and it triggers this umami taste, which is a very sought after taste in certain cuisines. We also have water receptors in the pharynx, in the throat. And water receptors are there to let us know when we have drunk enough water. So when we go back to our autonomic nervous system and we talk about our endocrine system, and we're trying to balance the fluids in our body and conserve water or get rid of water, then when we stimulate thirst, say we're low on body fluid and we need to generate more blood plasma volume, then we'll need to drink more water. So we have taste receptors that let us know when we have actually accomplished the task of drinking the water. All right, so gustatory discrimination is also going to be dissolved chemicals that are going to be contacting the taste hairs. Now, we've got different types of gustatory membrane receptors that will be operating. For example, we can have ion channels, and we can have G protein mediated receptors as well. So what we're going to see is we're going to have the receptor is going to release neurotransmitters when it is sufficiently stimulated and depolarized, and we will see that the neurons have their dendrites wrapped around the receptor membrane at the basal end. And so if the neurotransmitters are generating the action potential in the afferent fiber and they're able to depolarize it enough to threshold, then we will have a series of action potentials. So basically, taste sensitivity is also one of those things that can decline with age. The taste buds themselves can decline with age, and as we will see, people who eat lots of really hot and spicy foods or are constantly exposed to acids and things like this, they can deteriorate these taste buds' ability to regenerate fairly quicker. So a lot of people who have really grown up in cultures where you have really hot food, by the end of their lifetime, they need to eat really hot food in order to be able to taste it. All right, so here is our receptor cell. It's going to generate this generator potential here, and then that will cause the depolarization of the dendrite of the afferent fiber. And if we are able to depolarize it to threshold, then we will generate our action potentials. All right, now let's look at some of these channels. Some of the channels are thought to be ligand-gated channels for sodium and hydrogen and maybe potassium and other things. I'm not going to get too much into that. Some things can actually diffuse through leak channels. So sodium and hydrogen have been shown by some researchers that they can diffuse right on through leak channels, and they don't even have to open a channel through a ligand. They can just go right on through. The others, the sweet, bitter, and umami receptors, have been found to be G protein mediated. So once again, we have, in this case, we've got probably some kind of sugar molecule here. It's going to bind our membrane receptor, and then our peripheral membrane protein, our G protein, which in this case is going to be called a gustducin. It will initiate a cascade of second messengers.